Thanks so much for inviting me to talk about our work on the hyphosphere. First, I'm going to give you a quick overview, if I can get the clicker to work, of what I'm going to talk about this morning. First, AMF, what are they and why should you care? Next, I'll explain why the hyphosphere is a key soil microhabitat using 3 vignettes from research that I've either led or collaborated on. And then I'll spend the remainder of the talk talking about a new study where we used high throughput SIP to create a food web for the AMF hyphosphere. And so this is some new data, and I'm excited to share it with you today. It's been a fun data set to work on. So first, what are arbuscular mycorrhizal fungi? That's a mouthful. And so I'm a person who remembers words by breaking them down to their roots. So arb means arbor or tree. Myco means fungi, and rhiz means roots. So these are tree-like fungi that associate with roots. And if you look at the first fossils of land plants, you can see that they have these tree-like structures inside of them. So what they think is that this ancient symbiosis is actually what helped plants colonize land, and perhaps they served as early roots. And the function of this tree-like structure is to serve as nutrient exchange between the plant host and the fungal symbiont. So these are obligate symbionts. They're getting all of their carbon from the plant host. In return, they're giving nutrients like phosphorus and nitrogen back to the plant. And so what's really interesting about this, and it's something that has intrigued us over the years, is that AMF will actually, even though they can't decompose organic matter, they will go out into the soil and seek out bits of organic matter and can actually accelerate its decomposition. So how does this work? Do the AMF have something like a gut microbiome surrounding them, akin to our gut, where they're interacting with microbes to help do this decomposition? So this is a picture by Mary Firestone for our work, where you can see these AMF hyphae that are growing around this decomposing litter out in soil. And so AMF are not only, as I just said, are important for plant nutrition, and also, most plant species on Earth can form associations with them. They're a key predictor for soil carbon storage in terrestrial ecosystems. And then also to really hone in on this question of what's the importance of these AMF bacterial interactions. This is some work done by Rachel Hestrin, who is a postdoc in our group. And this is work she did as a PhD student, where it appears that AMF, in collaboration with their soil microbe, can take up a large amount of nitrogen derived from soil organic matter. And if you were to break any of these individual parts down, isolate them by themselves, they don't account for nearly as much nitrogen as you can account for when they're working together. And this synergistic interaction can contribute up to approximately, they're estimating about 70 teragrams of plant nitrogen annually. So now focusing down into the hyphosphere. So a lot of AMF studies have kind of focused on the root zone. So that's where the roots are interacting with these hyphae. But what we're going to talk about today is a zone away from the roots, out in the hyphosphere. And so what's the hyphosphere? It's the zone of interaction surrounding hyphae. So if you think of a rhizosphere, we typically think of that as a couple millimeters surrounding a root. But these are much smaller. These are structures that are only 1 to about 18 microns in diameter. So we're talking about the nanometers and the microns directly surrounding these hyphae. It's a very small zone. However, I think of this as a mighty microhabitat, because if you compare how much pore volume roots can explore compared to these hyphae, hyphae can explore, are estimated to explore about 200 times more pore volume than plant roots. So if you think of that in terms of carbon, that's an important movement of carbon out into the soil away from roots. But that's also an important foraging zone where hyphae can gather nutrients from areas that aren't depleted in the root zone and bring them back to the plant host. So for the first vignette, this was the first study that I worked on. And this was where we were trying to convince ourselves that can AMF access nitrogen from organic sources. And so what we did is we did an experiment, and a lot of the experiments I'll show you today kind of have this theme where we have a plant and a mycorrhizae growing on one side in a sterile media like sand, and then is separated by a filter that only those small little AMF can go through And then we put something delicious on the other side, say litter. And in this case, this litter is labeled with 13C and 15N. So that is a tracer to tell us, if we see 15N on this side, we know that the AMF picked up that nitrogen and brought it back to the host. So in this first image, I'm showing you some imaging mass spectrometry using nanosims, where we're looking at the root, that's this big blue background, and you can see that the hyphal filaments in that root are highly enriched with 15N. That's why they're glowing gold here. So we can see that, yes, the AMF brought 15N back to the plant host. And we can also see that the AMF significantly modified their microbiome while they were doing this. So all of these blue tips here are clades of bacteria that were significantly enriched in the presence of the AMF hyphae. In our next vignette, we were curious about how AMF bacterial interactions occur over time. And to be honest, what we were originally expecting to find here, so this is a network analysis, and we're looking over multiple weeks, and we're comparing this rhizosphere zone to what I'm calling a bulk soil zone out here. We were initially expecting to see, all the actions in the rhizosphere. That's where we're going to see all of these interactions. But what we were really surprised to see was that these AMF bacterial interactions were happening out here away in the bulk soil, and that these are increasing over time. So by the end of this study period, we have the most potential interactions with other microbes. And so, when we think about it, so what I was just telling you is, that root zone is nutrient depleted, and where we think all the action is happening in terms of where AMF would need to interact with their microbiome would be out beyond the root zone where they can access organic material. And the last quick vignette is a recent study where we were looking at how AMF can alter the physiology of their microbiome in the hyphosphere. And we were looking at this in a drought study. So this was a model bioenergy grass, Panicum pollyi, and we were looking under 3 treatments where we have an uninoculated condition and 2 different types of mycorrhizal fungi. And we had 2 different water conditions. We either watered it for 3 months, or for 3 months we gave it no water at all. And then we used 18O stable isotope probing as a metric of, after we resuscitate you, how much do you grow? And we can directly measure that by seeing how much of that isotope it takes up and brings into its DNA. So what you can see is when we gave it a lot of water, these microbes had a lot of growth potential. But in the in the drought condition, that activity was dropped. However, when we provided mycorrhizae, that actually essentially rescued activity so that in these drought conditions, they were able to perform similarly as they were in the water replete condition. So that is suggesting that these mycorrhizae may be supporting the resilience of the soil microbial community to moisture stress. Okay, so now I'm going to dive into this new research. And this is really going to go back to that initial question. So I hope I've showed you that we really think that the AMF are partnering with their microbiome to do this decomposition. But how does this work? And to do this, we're going to follow the carbon. And so this is, we did a natural labeling study. So we put grass into a 13 CO2 atmosphere. The plant fixes that carbon. It brings it down into its root. It hands it to the microbiome, and then we give it a little obstacle course where it has to jump over a little gap that's about 1 centimeter wide, and then it can get into the soil. And once it's over here, then we've isolated the hyphal carbon from the plant carbon. And then we can trace that carbon into a bunch of different pools by using CYP metagenomics. So some possible pools would be, microbes using AMF necromass, microbes predating upon living hyphae, maybe microbes that are using polysaccharides produced by the AMF, organisms consuming exudates, and then a whole suite of other organisms that could be using all of these types of carbon in addition to other carbon substrates, like normal litter out in soil. And then all of these organisms are producing carbon dioxide as they're respiring. And so that could fuel autotrophic growth of other organisms like ammonia oxidizers. So to give you a little image of what this soil looks like that we collected, this was done by Anne Kakouritis, and she did a water study that was recently published in New Phytologist. So this is our new carbon study. And so you can see we she collected these hyphae that are covered in soil. And so these plants were labeled with 13CO2 for 6 weeks. And then once we collected those, we extracted their DNA. We didn't get a ton of DNA, and that so we only got about 350 nanograms to work with. So for SIP, that's pretty low. But we had some important methodological improvements that allowed us to work with DNA that low. So Emily gave you a little overview of how SIP works. I'll just quickly run through it one more time. So in a SIP study, you think, you are what you eat. As you eat these isotopes, you put them into your biomass, you put them into your DNA, and that allows you to distinguish actively growing organisms from dormant or dead organisms. And so that enriched DNA literally becomes enmeshed in your background, the backbone of your DNA. And that gives us a neat property where we can then, that DNA becomes denser. We can put it in a density gradient, we can separate it out. In an ideal world, it would always look like this, and SIP would be so easy. We'd say, oh, look, we can just sequence this. We've got all the labeled populations. But typically, we more get something like a smear. So here we have this density, where we go from light to heavy density, and DNA on the y-axis concentration. And blue is our unlabeled group, and red is labeled. So you can see over here, we've really shifted the density over, and it's become highly labeled. But then we have this sort of gray zone of organisms that are partially labeled. And so in the past, the recent past, we would typically take just a few fractions and sequence them and say, this is the label zone right here. But that misses a whole bunch of different organisms. So SIP, low AT organisms tend to just be not as dense. Their DNA is not as dense. So then if you have an organism that is super AT rich, you would miss their DNA that became enriched. So the way that we are now moving is to sequence multiple fractions across the whole gradient. So in this study, we sequenced 14 metagenomes spanning the entire gradient. And then we used MetaHipmer to assemble those, all of our sequences together. And it did it in like a half hour. It was kind of freaky, actually. But and then using QSIP, we can estimate how much isotope did those organisms take up. And that allows us to start thinking about a food web, not just from a mag perspective, but also from an isotope perspective. How much carbon did you take up? What other types of carbon sources are you using? And so we did this, our SIP work, using a high-throughput pipeline that we developed as part of a JGI ETOP grant. This has now been implemented in the community science program, and you can apply to use this pipeline here. And so we used a couple of instruments. So we automated the fractionation, so you're not watching drips and seeing your life pass before your eyes. That's been a really big improvement. Automating, so that puts it into a 96-well plate. So then that's amenable to liquid handling, cleanup, and quantification. And just to give you a quick overview of how well this works, this paper is up on bioRxives if you want more details. So it really speeds it up. So it takes about half the time, and more importantly, about 1/6 of the hands-on labor. And you can see just from looking at manual versus automated fractionation of about 20 gradients each, that this really reduces the variance of these different gradients. And that one little technique that helped us work with only 350 nanograms of DNA is we added a teeny tiny amount of tween to our gradient buffer, and that about doubled our DNA recovery. So for working for small amounts of DNA, that could be a key development. Okay, so going back to our mycorrhizae. So now we're looking at our messy soil-covered hyphae again. Everything about this initial experiment looked like it was going to fail until we actually did it. So I want to walk you through that, because I feel like in a lot of SIP studies, we become discouraged by the amount of enrichment. But it's possible it might work if you're willing to sequence many, many fractions across your gradient. So this only had 1.8 atom percent 13C. So to give you a perspective, all of the 13C, natural 13C on Earth is about 1.1 atom percent. So that's barely a blip of, it's enriched, but not by very much. And we ran the gradients, and you can see I don't have this beautiful bump over here of enrichment, like it's maybe subtle, and you're like, maybe if I squint, look at these different curves, I can see maybe it enriched. But what gave us some confidence to move forward with this data set is that Anne had plucked out some of these hyphae and did nanosims imaging mass spectrometry on them. And she found that they were within a range of 12 atom percent to 61 atom percent enriched. So on average, 35 atom percent. So that's pretty screaming hot. So we were thinking, okay, maybe those nanometers or microns directly surrounding the root would also become enriched, and that we could still see it despite the fact that we have a very large 12C background. And now what I'm showing you, this is the AMF host, and you can see that this is clearly separated from the 12C. So we're like, oh, thank God, because that was a lot of money and effort, but it did work. And in fact, for the mags that we produced, approximately 212 of them had detectable 13C enrichment. And the groups that were most enriched were from the Myxococcota, the Fibrobacterota, the Verrucomicrobiota, and the Nitrososphaera. So that's an ammonia oxidizing archaea. So I'm going to tell you mostly about this group that is highly enriched, but we'll talk a little bit about these groups that are less enriched. So this is just plotting this in a different way. Here our dotted line is our AMF enrichment, where we have our organisms that are primarily using AMF carbon with this orange bar on top, and organisms that are using other carbon sources in addition to AMF carbon, and these are less enriched, so 5 to 10 atom percent. And so now we can use both this isotopic functional information alongside our MAG gene content in order to construct a hypothetical food web. So I'm going to just spend the rest of this time walking you through it, so you don't need to take this all in right now. So we'll first focus in a little bit and just look at our sources of isotope. So our primary producer, the plant, is about 41 atom percent. And so this AMF, when we look at by nanosims, we saw that that was a range that includes that number. But when we look at it with QSIP, it says it's 24 atom percent. So now we're going to look at, okay, who is our most enriched organism? That is this Myxococcota group. And it's a little bit higher than this 24 atom percent. So that's making us think that, okay, you are what you eat. Perhaps what they're eating are the fresher hyphal exudates or newer hyphae that was recently transported by the plant and out to the hyphae. And what's interesting about this group is that this is a group of known facultative predators. This particular class that we identified, so here I've grouped all of these by isotopic enrichment on top. And so you can see that all of these mags right here are from the Fen 1088 group. So we don't really know much about this group. There's maybe 30 mags in GTDB, and then all of those came from a peat metagenome. So this is a little known group, and so we were asking, okay, we don't know much about these. Are these predators? Because that would be, that'd be a great way to become enriched if you're eating the AMF carbon directly. And so in terms of comparing it to other Myxococcus type species, these have large genomes and a high GC content. So that's kind of classically like other Myxococcus. And another feature that they have is their genomes have a large arsenal of proteases. And so that's one way that, that Myxococcus attack their prey is by breaking down their proteins. And so they do have hundreds of proteases like other Myxococcus type species. But one thing's a little weird about this that the type species don't have is they have some genes for polysaccharides that are not well characterized. So it's hard to say exactly what they're doing, but they're for breaking down glycoproteins or fucose-based polysaccharides or some alginate-like compounds. And so AMF are known to produce a large amount of glycoproteins. People think that might be serve as a role in soil aggregation. Are they targeting those? That would be a great study. Somebody. We know that hyphae have fucose-based polysaccharides on their surfaces. This alginate-like compound, that is completely unknown, but that was really a gene that we found primarily in organisms that were highly enriched. So perhaps that gene is involved in targeting some type of fungal polysaccharide. Okay, so now we're going to move down to our next rung of our isotopic food web ladder. So we started from most highly enriched, and now we're going to go to an organism that had the same enrichment as the AMF. So again, if you are what you eat, you would think, okay, this is some either eating the hyphae or directly eating carbon that came from it. So this is a Fibrobacteriota. And before I show you another graph, I want to just quickly orient you to this graph up here. So we again we've ordered everything by isotopes. And now what we're doing is we're layering on top of that information about the MAGs. So we're saying, okay, I'm looking at your cazymes. What are the putative substrates you could potentially eat based on this gene content? And so based on that, one thing that is unique about this particular genome is it has a lot of chitinases compared to the other MAGs. So it has 11 putative chitinase genes. So if you know about fungi, you know that that chitin is part of the cell wall of fungi. And also that AMF turnover every 5 to 6 days. So if you think about, that's like a lot of dead biomass. So there's probably more dead AMF hyphae out there in soil than there is living hyphae. And recycling that could be potentially an important source of nitrogen to the system. And in terms of AMF, that's kind of a significant investment to lose every 5 to 6 days. So it's possible that this organism is performing a chitinolytic role, and that could help recycle AMF nitrogen. And then again, like what I said before, is these organisms also have some of these same polysaccharide genes for depolymerizing polysaccharides that the Myxococcus had. So perhaps these might be some AMF specific or fungal specific genes to break down polysaccharides. Okay, now we're moving again down in our ladder. At this point, we're now at about half of the enrichment that we started off with here. And so this is a Verrucomicrobiota. And so it's getting approximately assimilating about half of its carbon from the AMF and half of it from somewhere else. Is that soil organic matter? Is that DOC? It could be multiple different sources. What leads us to think that it might in partly relate to soil organic matter is that this organism has more genes to degrade polysaccharides than some of its other highly enriched organisms. So that's this gray bar. And also that this family is typically found in rice paddies or insect guts or areas where you'd think you might have some decomposition going on. Another thing that was interesting is I did a metatranscriptomic study a couple years ago. And this was one of the few groups I found where when we looked at expression of decomposition genes in the rhizosphere alone and the detritosphere alone, when we brought them together, they had a synergistic expression. So they were really excited and turning on their genes when they had both exudates and litter together. So that suggests it could be SOM influenced decomposition. And then if we look further down this food web, you can see that in some of the organisms that are less enriched, they also have a very high potential for decomposing things. So, relating back to our initial question, are the AMF partnering with their microbiome to break down organic matter? These would be my most wanted list. I would guess if we were to look at this, with transcriptomics or follow it up with an activity study where they have a high potential to do decomposition. And now we're going to move to the center of our food web. So this is a Nitrososphaera that also had about half the enrichment of the AMF. So these organisms are thought to be primarily autotrophic. So it could be that they're using the CO2 produced by the other organisms in the community to become enriched. Another route that they possibly could become enriched by is that this MAG, like a lot of other Nitrososphaera MAGs, has the genes to make a urease. So that's where you break urea down into CO2 and ammonia. So that could support both 13CO2 uptake and also respiration. And there is some preliminary evidence that it appears that AOA prefer to use nitrogen from organic sources, ammonia derived from organic sources. So that could be a possible mechanism to get labeled. Another Also, people are currently investigating if they can do mixotrophy. So I just put that in, who knows, maybe they could be. Okay, so now we've completed this loop, and I've layered on top of this the different nutrients that could be generated by these processes. These are things that the AOA that are living could potentially take up. It's also a potential point where AOA could be competing with the AMF for, say, ammonia that's produced. So this is kind of an intriguing thing that we didn't expect to see in the middle of our food web. Let's say to quickly show you what's not in here. So exudates are really hard to identify what's breaking those down. You don't need a fancy depolymerase genes to break down like a monomer. So it's doing something like metabolomics may be a better way to investigate that particular question. Also, SIP tends to, or metagenomics tends to assemble things that are super abundant and prokaryotic. So we're missing rare things. I'm really interested to hear more about the eukaryotic binning because I'd love to get the eukaryotes out of the system. Though we're planning to maybe take a transcriptomic approach to help answer that question. And then finally, viruses. So there's exciting things to come. There's a lot we still have to learn. So this seems pretty complicated, but it's really just scratching the surface of this little food web. And finally wanted to reiterate, these are our genomic potential. We really need to do activity in order to demonstrate what's happening here and what genes are used and where they're used. So in summary, the hyphosphere is a mighty microhabitat. Using high throughput stable isotope probing, we identified some possible roles that these MAGs could be performing in this environment. And that isotope really gave us a way to identify organisms that are using the AMF carbon and gave us some nice hypotheses about what types of carbon sources they're using in the system. And these MAGs that we developed are a genomic resource that can support future activity-based measurements. So all of this work is up on bioRxiv. I just got the first round of reviews from our paper at Microbiome, and it looks good. So hopefully it's coming out soon in its final form. And with that, I just wanted to thank Jennifer Petridge and the SIP team at Livermore, our bioenergy SFA, my DOE early career, Mary Firestone, who I've collaborated with for a long time at UC Berkeley, and then our collaborators at JGI, and you for your attention. Thank you.